Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 1 for October 2022. Let us begin with the first question. Question 1. The cell membrane controls which substances can enter and leave a cell. The cell membrane is a phospholipid bilayer with proteins embedded in it. The shapes show the components of a phospholipid. So we have the phosphate group, we have the glycerol, the fatty acid, as well as a covalent bond. And they say, draw a diagram to show the structure of a phospholipid using these shapes. We know that a phospholipid contains two fatty acid tails. It contains one glycerol, as well as one phosphate group. I drew the covalent bond slanting in a similar way that the question gave us. So my structure was as you can see here. Moving on. Here they say, which type of bond is found in this molecule? The molecule has an ester bond, so the answer should be A. Part B says, how does a nonpolar molecule move through a cell membrane down a concentration gradient in a cell? A nonpolar molecule will move through the cell membrane by diffusion, so the answer here should be a B. Next they say, how many of the following statements are correct for the transport of a substance by endocytosis? Transport substances out of a cell. That is wrong because endo means into, so taking inside the cell. The cell membrane surrounds the substance. That is correct. And can only transport substances down a concentration gradient. This is not correct. So there is only one correct statement, and the answer here should be a B. So this brings us to the end of question 1. Let's continue to question 2. Question 2. The diagram shows the time taken in seconds for an impulse to travel through different parts of the heart. So down here they say, what is the name of blood vessel Q? Blood vessel Q is the vena cava, and therefore my answer should be a D. Next they say, the cardiac cycle is divided into 3 stages. 2 of these stages are arteriosystole and ventricular systole. Name the other stage of the cardiac cycle. The answer I wrote here was cardiac diastole. Moving on. Here they say the duration of ventricular systole is the time it takes for the impulse to pass between E and F, shown in the diagram. Calculate the proportion of time that the ventricles are contracting in one heartbeat if these heartbeats 73 times per minute. So moving back to C, E to F. Moving E to F, you can see this is what we have, and it's 0.05 seconds of the total time. So the total time is, of course, that plus that plus that plus that plus that. So 0.05 is what it takes for contraction of the ventricles to occur. So since they've told us there's 73 beats per minute, we can say 73 beats in 1 minute, which is 60 seconds. And therefore 1 beat or 1 heartbeat, it's going to equal to 60 divided by 73, which gives us this time. And then for ventricular contraction, moving from E to F, we have 0.05 seconds. So the proportion is going to be that divided by that, which gives us 0.0608, which is 0.06. And that is my answer. Next they say, the part labeled B is in the wall of the atrium, and the part labeled D is in the wall of the ventricles. The distance between B and D is 12.5 millimeters. Calculate the speed at which the impulse travels between B and D. If we go back B to D, moving from B to D, we can see it's 0.12 seconds. So that is the time I wrote here. So it means we are moving this distance within that time, and therefore the speed, which is distance over time, is going to be that distance divided by the time. So that was my answer. Next they say, explain why this speed needs to be slower than the speed that the impulse travels between other parts of the heart. The speed needs to be slower so that we allow the atria to fully empty and then the atrioventricular valves to close before the impulse can travel to the ventricles to cause ventricular systole. So I said the speed should be slower to allow the impulse to move across the septum around the atrioventricular region. And this allows the atria to be fully emptied so that the atrioventricular valves are fully closed before the ventricles can contract. So this brings us to the end of question 2. Let's continue to question 3. Question 3. The structure of blood vessels relates to their function. Describe how the structure of capillaries relates to their function. Capillaries are 1 cell thick on the side. It's like 1 cell, and then this side is 1 cell. So the diffusion distance is going to be shorter. Also, they contain pores that will allow for effective diffusion of only small substances meaning the bigger molecules cannot cross from the capillaries into the tissues surrounding the blood vessels. Next they say, draw a cross-section of an artery to show the details of its structure. Label two features of the artery on your diagram. So here I labeled the lumen as well as the endothelial layer. Of course, we have the inner layer as well as the outer. So if you could draw a list and label two parts like I did, you would get 3 marks that were awarded. Next they say, explain why veins need valves along their length. We need to remember that blood flows through the veins at a lower pressure, yet veins are returning blood back to the heart. Therefore, valves are required in order to prevent the backflow of blood. So I said, to prevent the backflow of blood due to gravity, since blood in the veins is at a lower pressure. Moving on. Here they say, deoxygenated blood is taken to the lungs and oxygenated blood is taken away from the lungs. Complete the graph to show the changes in partial pressure of oxygen in the blood as it flows back to the lungs, through the lungs, and away from the lungs. Down here on the vertical axis, we have partial pressure of oxygen in the blood, and then on the horizontal, we have the direction of flow. So when blood is flowing back to the lungs, of course, there is going to be a very slight decrease. Actually, you could have drawn this being a flat line. It could be that it doesn't have any change, but because within the lungs there are cells, we expect some respiration to occur, and a very tiny decrease can occur. So I drew a slanting decrease, which is quite small. And then here, as blood flows through the capillaries of the lungs, we expect a very big increase in partial pressure of oxygen within the blood. And then as blood flows away from the lungs, we expect a slight decrease in the partial pressure of oxygen. So the key thing is at least you have to draw something within one big square here, and one big square here. This can be as big as you want it. So this brings us to the end of question 3. Let's continue to question 4. Question 4. Enzymes are biological catalysts that have an effect on the energy required for a reaction. State the meaning of the term biological catalyst. The term biological means they work in organisms, and enzymes are catalysts because they decrease activation energy for reactions, hence speeding up the rate of reaction. Moving down here, the graph shows energy changes during an enzyme catalyzed reaction. On the vertical axis, we see energy, and on the horizontal axis, we see time. And they ask which arrow shows the energy required to start the reaction. The energy required to start the reaction is going to be Q because that is the activation energy for that specific reaction. So my answer here was B. Moving on. In part C, the diagram shows a series of chemical reactions. So here we see A plus B catalyzed by enzyme 1 to give us AB, which is catalyzed by enzyme 2 to give us AC. Again, AC catalyzed by enzyme E3 to give us D. So they say 3 different enzymes, E1, E2, and E3 are involved in these reactions. Explain why E1 cannot convert AB into AC or AC into D. Of course, this is because enzymes are specific in nature because they have specific active sites that can be complementary to a specific substrate. So the enzyme substrate complex cannot be formed between E1 and AB or E1 and AC because they're not complementary. So I said, enzyme 1 is a protein with particular R groups enabling formation of active sites. These active sites have a specific shape, and this shape is not complementary to AB or AC as a substrate, or you say as substrates, to form an enzyme substrate complex. So moving on. Here they say, pectinase is an enzyme that breaks down pectin. The effect of different combinations of pH and temperature on pectinase activity was investigated. The volume and concentration of pectinase and pectin were kept the same in all reactions. The table shows the rate of reaction for each pH and temperature combinations tested. So we can see they tested pH 2, and then pH 4 and pH 8. We also see temperature 5, 30, 60, then 530, 60, and 530, 60. So for each pH, they changed the temperature, and for each temperature, the pH was varied. The rate of reaction is 0 here, and 0 there, but we see the highest rate is here, meaning temperature 30 and the pH 4 are going to be the optimum conditions for this reaction. However, down here we see the optimum pH, but at a higher temperature, and the rate was 0. This is because at this temperature, the enzymes are completely denatured. And up here we see at the perfect temperature, like we saw here, which is optimum, when the pH is 2, again the rate of reaction is 0, and that is because at pH 2, there is going to be denaturation of the enzymes. So down here they say, describe two conclusions that can be drawn from these results. I said, from these results, the optimum pH is pH 4 and the optimum temperature is 30 degrees. We can see that here. These are the optimum conditions because they had the highest rate. And then the temperature, 60 degrees, and pH 2 resulted in the lowest rate because they denatured the enzymes. So this brings us to the end of question 4. Let's continue to question 5. Question 5. Gas exchange requires diffusion. The rate of diffusion is dependent on the surface area, the diffusion distance, and the concentration gradient. The rate of diffusion can be calculated using the formula, which we see here. Rate of diffusion is proportional to the surface area times concentration gradient divided by the diffusion distance. They say, name the law on which this formula is based. This formula is based on fixed law of diffusion. Of course, greater surface area, greater concentration gradient, lower diffusion distance is proportional to a higher rate of diffusion. So here they ask, what will happen to the rate of diffusion if all three of the following are changed at the same time? If the surface area is halved, the concentration gradient is halved, and the diffusion distance is halved, what changes are we going to see? So I rewrote the same expression, but in this case, I used half the surface area, half the concentration gradient, as you can see here, and half the diffusion distance. We can see the half cancelled out, and then we remained with a half times surface area times concentration gradient times diffusion distance. And whatever we get here is half of what we have here, meaning the rate is also going to be half. So my answer was C, it will be half. Moving on. So here they say, the properties of gas exchange surfaces include a large surface area, a short diffusion distance, and a high concentration gradient. Describe how mammalian lungs are adapted to have each of these three properties. Now, the mammalian lungs have many alveoli, and these provide a large surface area. Also, the walls of the alveoli are 1 cell thick, meaning they provide a shorter diffusion distance. They also have numerous capillaries, which is a greater blood supply to provide a higher concentration gradient. In this case, as oxygen comes in, it is going to be taken away immediately due to the many blood capillaries that are present around the alveoli. Moving on. Here they say, the photograph shows a hellbender salamander. So this is the salamander. They have labeled the skin folds. And down here they say, these salamanders live in freshwater streams that are shallow and fast-flowing. They use their lungs and the surface of their skin for gas exchange. They also use body rocking and swaying movements in water. The graph shows the relationship between the frequency of body rocking movements, blood partial pressure of oxygen, and the percentage saturation of oxygen of the water they live in. If you can see, everything I underlined is important in the questions that we're going to answer. So here we have the graph, the frequency of the body rocks on the vertical axis. On the horizontal axis, we have percentage saturation of water with oxygen. We have results for partial pressure of oxygen and results for frequency. So we can see at lower percentage saturation of water with oxygen, we see the frequency of the body rocking movements will increase until a specific point, and then they will decrease. The decrease could be because they have enough oxygen in the water, and also the salamanders are tired, so they need to conserve the energy for other body processes. So let's continue to the next question. Here they say, discuss how the structure and behavior of these salamanders are adapted for gas exchange. So I want to take you back here. I'm going to go through the properties I underlined here. They say the salamanders live in freshwater streams that are shallow and fast-flowing. If they're fast-flowing, it means it allows for more oxygen dissolution. If there is more oxygen in the water, greater diffusion is going to occur through the skin of the salamander. The shallow water allows the salamander to be able to access the air so that it can be able to use its lungs to breathe in order to take in more oxygen. And again, here they say they use their lungs as well as the surface of their skin for gaseous exchange. We can see the folds allow for greater gaseous exchange, or they provide a greater surface area for gaseous exchange. Down here they say body rocking and swaying movements. These are agitating the water in order for more oxygen to be dissolved so that they can have more oxygen closer to the surface of their skin in the water in order for more diffusion to occur. So taking you back here, I said the salamanders use both their skin and lungs for gaseous exchange. Their skin is permeable to gases, meaning even to oxygen. The lungs enable them to get oxygen from air, and their skins allow them to obtain oxygen from water. Their skin is folded, and some parts of the body are flattened to provide greater surface area for the diffusion of oxygen. Salamanders live in shallow, fast-flowing water. Fast-flowing water contains more dissolved oxygen, and shallow water allows them to breathe oxygen from the air, meaning using their lungs. The rocking and swaying movements agitate the water to promote more oxygen dissolution so that their skins are in contact with more oxygenated water so that more oxygen diffuses through their skins. From the graph, there are more rocking frequencies at low percentage saturation of water with oxygen, followed by an increase in the blood partial pressure with oxygen. The rocking movements decrease when water saturation with oxygen increases. Also, rockings decrease to conserve energy. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Warfarin is an anticoagulant. The dose of warfarin that is given to a patient has to be monitored regularly. This is done by adding thromboplastin to a blood sample and measuring the time it takes for the blood to clot. The result is compared with a contrast sample and is reported as an international normalized ratio, INR level. How many of the following statements are correct for thromboplastin? It is stored in platelets, that is correct. It is an active enzyme, that is correct. It is converted into thrombin, that is wrong. Prothrombin is converted into thrombin using the active enzyme thromboplastin. So we have only 2 statements that are correct, so my answer is a C. In part B, the graph shows the relationship between INR level and the risk of stroke and the risk of bleeding within the skull. So on the vertical axis we have the risk, and on the horizontal axis we have the range of INR. So we can see the risk of stroke decreases, and actually it has a greater decrease in comparison to the risk of bleeding within the skull. Then a fluctuation about these ranges before we see an increase in the risk of bleeding within the skull and a slight increase in the risk of stroke. This seems to be the range where we have the lowest risk for bleeding in the skull, and this seems to be the lowest risk of stroke. Moving on. Here they ask, did you use a safe range for the INR level? Use the information in the graph to support your answer. Here I chose the range between 2.0 and 2.5, if we can see here, because for this range, both risks had lower values. So I said both have lower combined risks, although the lowest risk of stroke is at 3.1 to 3.5, and the lowest risk of bleeding in the skull was 1.4 to 1.7. Actually, acceptable ranges are 1.8 to 3.5 as well as 1.8 to 2.5. Down here they say, explain how taking this anticoagulant can reduce the risk of cardiovascular disease, which is CVD. So warfarin is an anticoagulant, so it's going to reduce the thrombosis or formation of blood clots. So that means blood clots cannot block the coronary artery. So blood with sufficient oxygen and nutrients will be delivered to the heart muscle in order for aerobic respiration to take place. Moving on. Here they say, warfarin is given to patients who have had a heart attack to prevent further heart attacks and other related health issues. A study compared warfarin with the use of aspirin over a 4-year period following the first heart attack. A group of 1,216 were given warfarin. 83.3% of these people had no further health issues. Calculate the number of people given warfarin who had no further health issues following their first heart attack. So the percentage is 83.3%, which is 83.3 divided by 100 times the total number of people, and this gave me 1012.9. But human beings or people cannot be in fractions, so I rounded this off to 1013, although you could leave your answer as 1012 as well. Moving down here, a second study compared this result with people given a combination of warfarin and aspirin. Describe how this study would be carried out to produce a valid comparison. For comparison to be valid, you have to use people who have had a heart attack as well. These people have to be of the same age, same gender, and have similar lifestyles as the people from the first group. Each person should be given the same quantity of warfarin as given in the first study, and each person should be monitored over a period of 4 years, or you could say over a long time. Here I can say 4 years. They should monitor them for a period of 4 years like they did to the first group. And then suitable statistical tests can be used to make conclusions on the results that have been obtained. So this brings us to the end of question 6. Let's continue to question 7. Question 7. Insulin is a small protein composed of 2 peptide chains, the A chain and the B chain. Insulin is made from the protein proinsulin. Proinsulin is composed of the A chain and the B chain joined by a sequence of amino acids called the connecting region. The diagram shows how insulin is formed from proinsulin. So here we have proinsulin. Ignore the things I wrote in blue because they helped me to answer the question below. And then from proinsulin we can form this insulin. That means the connecting region has been cut out, this part here and that part here, to create the insulin molecule. And then here down they say the connecting region is removed from proinsulin, leaving the A chain connected to the B chain by disulfide bridges. Which row of the table shows the position of an amino group and a carboxylic group? Now if you go back here, this is an amino group, and that means this is going to be the amino group. And then if this is the amino group, it means we're going to have the carboxylic group, which is here. And for the carboxylic group to be here, it means the connecting region is going to have an amino group here and a carboxylic group here. And that means this is going to have an amino group, and therefore that is a carboxylic group. So here, the amino group is going to be at position R, and then the carboxyl group is going to be at position P. So A is my answer. Moving on. Here they say proinsulin is composed of 84 amino acids. Insulin is composed of 51 amino acids. What percentage of proinsulin is the connecting region? The number of amino acids in the connecting region are going to be 84 minus 51, which gives us 33 amino acids. And therefore the percentage is going to be 33 divided by those in the proinsulin, which are 84, times 100, and I got 39.3, which is my answer here. Next they say there are 3 disulfide bridges in insulin. Each disulfide bridge is formed between the R groups of 2 cysteine amino acids. Give the ratio of amino acids forming disulfide bridges in insulin to those not forming disulfide bridges in insulin. Since disulfide bridges are formed between 2 amino acids, and there are 3 disulfide bridges, it means 6 amino acids are forming the total of 3 disulfide bridges. So of the 51 amino acids, 6 of them are forming the disulfide bridges, and 51 minus 6 gives us 45 that are not forming. So the ratio is going to be 6 to 45, which is 0.13 to 1, or you could say 1 to 7.5. So either this would be a correct answer. Then down here, suggest how insulin is synthesized from proinsulin, and then use the information in the diagram to support your answer. If we go back here, making insulin from proinsulin, since we cut out the connecting region, which is composed of many amino acids, the total is 33, it means there is a peptide bond that is going to be broken here, and another peptide bond that is going to be broken here. To do that, we need enzymes to carry out hydrolysis reactions. So here I said, enzymes carry out hydrolysis reactions. The peptide bonds between the connecting chain with position P and position R are broken. Moving on. In part D, insulin lowers the level of glucose in the blood. This glucose is taken up by the liver and stored as glycogen. Compare and contrast the structure of glucose and the structure of glycogen. Now, when they say compare and contrast, of course, you have to write the similarities as well as the differences. For similarities, both are made up of alpha glucose. Also, both contain 3 atoms, which are carbon, hydrogen, as well as oxygen only. The differences are glycogen contains 1,4 and 1,6 glycosidic bonds, while glucose does not. Also, glucose is a monosaccharide, while glycogen is a polysaccharide. Moving on. In part E, type 2 diabetes is a disorder that can result from ineffective or insufficient insulin. It is a risk factor for cardiovascular disease. Cinnamon is a spice that has been shown to increase the uptake of glucose from the blood into the liver, increase the synthesis of glycogen, In a study using rats, the effects of a cinnamon extract on levels of glucose in the blood was determined. In this study, a group of healthy rats and a group of rats with induced diabetes were used. The rats were given either cinnamon extracts or water each day for 30 days. The table shows the results of this study. So for healthy rats that are given water, we can see the level of glucose in the blood is decreasing, but the decrease is not that big. Also for healthy rats that are given cinnamon extract, we see the level of glucose is decreasing. However, when we look at the diabetic rats, those that were given water, we see there is going to be an increase in the blood glucose concentration due to the fact that they cannot produce insulin. And then the diabetic rats that were given cinnamon extract, there was a slight decrease in the concentration of glucose in the blood. So here they say, explain the results of this study. Since I've already explained from the table, I'm just going to go through the answers I wrote here. So I said for healthy rats given water, the blood glucose decreased slightly. Since they are healthy, they can produce insulin to control blood glucose levels. And for healthy rats given cinnamon, the blood glucose levels decreased slightly. Since the rats are healthy, they can produce insulin. Addition of cinnamon gave a combined effect for controlling blood glucose levels. For diabetic rats given water, they had a higher concentration of blood glucose compared to healthy rats given water because they could not produce insulin to control the blood glucose. And for diabetic rats given cinnamon, the levels of blood glucose were fairly maintained in a range but higher than the concentration of healthy rats. Cinnamon lowered blood glucose by moving it to the liver. It stimulated enzymes to convert glucose into glycogen. Cinnamon, however, did not stimulate as many enzymes as insulin does, so glucose levels did not completely decrease, but fluctuated within a range. So this brings us to the end of question 7. Let's continue to question 8. Question 8. Mononucleotides are joined together to form DNA. Each mononucleotide is composed of a deoxyribose linked to a phosphate and one of 4 bases. The diagram shows the structure of the 4 bases. Guanine and adenine have 2 ring structures. Cytosine and thymine have 1 ring structure. So, we have the guanine, cytosine, adenine, as well as thymine. They say explain the role of these bases in the structure of DNA. They want you to use the information in the diagram to support your answer. We know that these are used to form polynucleotide strands that pair up to form hydrogen bonds between complementary bases to hold the 2 DNA strands together. So the large purine bases, like adenine and guanine, will pair with smaller pyrimidine bases, like cytosine and thymine, forming the parallel DNA strands. So moving on. Here they say the DNA of the bacterium E. coli has a mass of 3.1 times 10 power 9 daltons and consists of 4.7 times 10 power 6 base pairs. Calculate the mean mass of a base pair and express your answer in standard form. So here we have 3.1 times 10 power 9, which is the total mass, divided by the number of base pairs, which is that. We get 6.5957 times 10 power 2, which I rounded off to 6.6 times 10 power 2 daltons. Here they say, calculate the total mass of DNA produced after 3 cell divisions and give your answer in grams, where 1 dalton is 1.67 times 10 power negative 24 grams. Since we had 3 cell divisions, each cell division results in doubling the total mass. So it means if there are 3 cell divisions, the mass is going to be 2 times 2 times 2, which is 8 times. So total mass should be 3.1 times 10 power 9 times 8, which gives us 24.8 times 10 power 9 daltons. Since 1 dalton is 1.67 times 10 power negative 24 grams, so we multiply that with the equivalent of 1 dalton to give us this in grams. Down here they say, Matheson and Stoll provided data that supported the theory that DNA replicates by the semi-conservative replication and disproved competing theories. Heavy nitrogen and light nitrogen were used in the experiments. The flowchart summarizes what experiment performed by Matheson and Stoll. In stage 1, bacteria was grown for several generations in culture medium containing the heavy nitrogen. And in stage 2, bacteria from stage 1 grown for 1 generation in the culture medium containing the light nitrogen. And in stage 3, bacteria from stage 2 grown for 1 generation in the culture media containing the light nitrogen. So after each stage, a sample of DNA was taken from the bacteria and the DNA molecules separated on a density gradient in a tube. The heavier DNA molecules form bands lower down the gradient than the lighter DNA molecules. The width of each band is proportional to the percentage of molecules in the sample. This is very important. They want you to complete the diagram to show the results of this investigation and use the dotted lines to help you position the bands on the diagram. The first one has been done for you. So in the first experiment, we only had the heavy nitrogen. So 100% is going to be the heavier side, and you can see everything is here. In the second experiment, this separates. Each pairs with the lighter nitrogen producing 100% mixed DNA, which creates this we see here, which is going to be in the middle. And then in the next experiment, each of these is going to separate like you see I did here, and each separated one is going to pair with a new strand composed of only nitrogen 14, which is this one, with 14, that with 14, that with 14, and that with 14, creating 50% light and 50% mix, and therefore those should be the bands that we see. Moving on. One competing theory was the conservative theory. The diagram shows the conservative theory for the replication of DNA. Here we can see 100% heavy, and then we see 100% heavy, and the other is going to be 100% light. So they say complete the diagram to show the position of the bands on the density gradient in a tube if this was the correct theory. If this is the correct theory, then we're going to have, like I said, 100% light and 100% heavy. There is no mix, so we're going to see of this total, we'll see the percentage is going to be that as well as that. The percentages, which is 50-50, if you add them together, they create 100%, which will be the same size. So 50 here and 50 there. Moving on. Another competing theory was the dispersive theory. The diagram shows the dispersive theory. Here we have 100% heavy, and then we can see 100% mix. So down here they say complete the diagram to show the position of the bands and the density gradient in a tube if this was the correct theory. Since we now have 100% mix, it means we're going to have the same size of the band but located in the middle because it's mixed DNA. So 100% mix is going to be represented as that. So this brings us to the end of question 8. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.